Good evening and welcome to the 59th Faculty Lectureship at NDSU. My name is Bruce Malath, Professor of English and Director of the Upper Division Writing Program at NDSU. On behalf of the Faculty Awards and Recognition Committee, I would like to welcome relatives and friends of Dr. Katti and all members of the NDSU community, President Dean Bershani, Provost Margaret Fitzgerald, faculty, staff, and students. Welcome also to faculty and friends from Concordia College and Minnesota State University Moorhead, and from across Fargo-Moorhead. This year, the committee has recognized Dr. Dinesh Katti as the recipient of the Faculty Lectureship, one of the most prestigious of all awards at NDSU. It recognizes stellar achievement in the three areas of teaching, scholarship or creative activity, and service. Dr. Katti has achieved impressive accomplishments in each of these areas, and you will hear more about them in a few moments. Dr. Katti is the most recent of more than 50 recipients among NDSU faculty dating back to 1957, when Professor W.C. Whitman spoke about grass on the prairies. Since then, distinguished awardees have spoken on such varied topics as Arctic exploration, atoms and energy, quantum chemistry, climate change, the geology under Fargo, fish and frogs, wilderness, what history tells us, teaching and learning, the conductor's art, and how we connect or not. Following Dr. Katti's talk, there will be time for questions via the chat function in Zoom. Please type your questions during the talk. I'll then sift through them and read aloud those that we can fit into the time allowed. After that, President Bershani will present the Faculty Lectureship Award. Following the ceremony, I hope we will send congratulations to Dr. Katti via the chat and Zoom and via email messages. I'll now turn it over to Dr. Michael Kessler, Dean of the College of Engineering, who will introduce this year's Faculty Lectureship, Materials Engineered Byte by Byte.
Thank you, Bruce, and thank you everyone for joining us this evening. Dinesh, it's good to see you there, and we're really excited to welcome you as this year's faculty lecture winner and presenter. For the audience, some things about Dinesh. Dr. Katti is a professor of civil and environmental engineering at NDSU, and he teaches geotechnical courses, including soil mechanics and multi-scale modeling. Professor Katti pioneered the use of computational multi-scale modeling and mechanics to explore nanocomposite materials to identify the key microstructural, nanostructural, and molecular scale nuances that result in unique properties exhibited by nanocomposite materials. These include biological materials, seashells, bones, cancer cells, geologic materials, swelling clays, and I'm sure we're going to hear a lot more about some of those things tonight. His work has led to important discoveries and has made a far-reaching contribution to the field. He developed powerful simulation tools to design materials with tailored properties, and he's a leading expert in the world in research of engineering behavior of swelling clays, where he has made important contributions to describing the mechanisms of swelling in clays and the resulting impact on infrastructure.
Dr. Kessler, I think you're muted.
Oh my, I'm so sorry. I just gave a great introduction to you, Dinesh. Let me start over, and thank you for interrupting me there.
Mike, I think we heard about half of it.
Yeah.
Oh, okay. Well, I'm not sure where I was, but let me just say a few things about Dinesh's awards. He's won just a great number of awards, including the John Brooker Excellence Award by the International Association for Computer Methods and Advances in Geomechanics, the Patriot Award from the Office of the Science of the Secretary of Defense, and NDSU's Fred Waldron Research Award, and the College of Engineering and Architecture Researcher of the Year Award. On a more personal note, I first got to know Dinesh when I came here in 2017. He was serving a very long interim as the department chair of civil and environmental engineering, and he was very welcoming and helpful in that transition, as well as continuing to be a world-class researcher. So I think we all want to hear from Dinesh, so I'll turn it over to you. Thank you.
Well, good evening, everybody, and thank you very much, Dean Kessler, for the nice introduction. Thank you, President Brishani and Provost Fitzgerald, for joining in person. It's a great honor and pleasure to give this 59th Faculty Lectureship Award presentation. I would like to first thank Dean Mike Kessler and Chair David Stewart for nominating me. I would also like to thank the Awards Committee for selecting me for this award. To my colleagues in the Department of Civil and Environmental Engineering, thank you very much for your support. I would like to acknowledge my students, past and present, whose dedication to science and hard work has led to many discoveries and major contributions. On a personal note, one person in my life gets credit for putting up with my nerdiness. That is my beautiful and smart wife, Kalpana, who's sitting here. She's not only my life partner, but also a research collaborator and recipient of this very faculty lectureship award a few years ago. She has been a strong motivator and an honest critique, sometimes too honest. Also, thanks to our daughter Priya, who thinks her nerdy parents are cool. I would like to thank all of you joining in remotely from around the world. If it were not for COVID-19, we would be here together in this beautiful auditorium. Well, I'm a civil engineer. And as a civil engineer, we design structures using computer simulations. And once the structures are designed, we go ahead and build them. There is no trial and error. So these are some of the projects I have worked on as a geotechnical engineer before coming to NDSU. On the other hand, materials are not designed using computers. Materials are typically designed by trial and error. And the problem with trial and error is that it is extremely expensive, it is time consuming, and it is difficult or almost impossible to design materials with tailored properties. So in our group, what we have done is we have introduced computer simulations to explore and model materials and design new materials. So today, I'm going to tell you a tale of 4 nanomaterials. And these 4 nanomaterials are nacre and bone. These 2 materials are of biological origin. Swelling clay, which is a material of geologic origin, and then polymer clay nanocomposites, which are synthetic nanocomposite materials. So let's see first what is nano. Nano is prefix for billionth. So nanometer is a billionth of a meter, or 10 to power minus 9 meters. I am, as a reference, I am 1.67 billion nanometers tall. And the size of water molecules is about 0.28 nanometers. So nanometer is really very, very small. Now, nanocomposite materials are when one of the constituents of the materials has a size of tenths of nanometers or less. And I'll be referring to this word called modulus or elastic modulus during my presentation. What it simply means, it just reflects stiffness of the material. If that number is big, then it is a very stiff material. The number is small, it is not so stiff. Now, these are some of the equations we have used in our computer simulations. Now, I do not want to bore you, so this is the only slide with mathematical equations that you're going to see. There are going to be no mathematical equations in subsequent slides. So the first nanoscale material or the nanocomposite material I'm going to talk is nacre, which is the iridescent inner layer of seashells. Now, seashells are these wonderful armor materials that protect the organisms that reside within them. The outer region of the seashell is called is a calcitic mineral, and this mineral or the calcite is basically very strong and very brittle or very hard. On the other hand, the inner layer of seashell, which is called nacre, is strong and tough. And now material scientists have been interested in nacre for a very long time. So the composition of a nacre is 95% aragonite. Aragonite is a calcium mineral similar to chalk. And the remaining part of the nacre are organics. It's basically a cocktail of proteins. But what is very interesting about nacre is that the fracture toughness or the resistance to fracture is 3,000 times greater than that of pure aragonite, that's the parent material. So although the material is 95% aragonite or something similar to chalk, the resistance to fracture of this nacre is 3,000 times more than the parent material. And that is incredible. And if we are able to find the reason why nacre has such high fracture toughness, we will be able to mimic the structure and make new materials which are thousands of times stronger than the materials we currently know. And so, and that's what materials, that is the motivation for this work. Now let's take a look at the microstructure of nacre. So on the right-hand side, what you see is what it looks like is a brick wall. And so the bricks are about 500 nanometers tall, and the mortar in between the bricks, that's the organic phase, and that is about 20 nanometers thick, making nacre a nanocomposite material. Now, in 1997, Kalpana and I were sitting and having lunch, and we were discussing about what are the new opportunities in the material science area, what are the questions that have not been answered in the material science area. And nacre seemed to be very interesting because people had worked on nacre for about 30 years and they had not found the reason why the nacre has this high resistance to fracture. And so we decided to use a radically different approach and use computations and mechanics to explore nacre and find and delineate the reasons or the structural reasons why nacre has such amazing fracture toughness or resistance to fracture. And so the first thing we did was to build a structural model of nacre down to the nanometer scale. And then we used, at that time, some of the world's fastest supercomputers to do our simulations. And at that time, we did not know what the elastic modulus of that organic phase, the 20 nanometer thick organic phase was. And our simulation showed that the elastic modulus of this organic phase is about 20 gigapascals. Now, that number, 20 gigapascals, is about 1,000 times more than what we were expecting. And the reason why I'm saying 1,000 times more, because this organic mortar is made up of proteins. And if you look at the bulk proteins, they have elastic modulus of only 20 to 50 megapascals. That's 1,000 times less than this number. Well, the first thing, of course, we did was to go and check all our numbers, whether our numbers were correct. And we were very confident that our numbers were right. And then we went ahead and presented this work in the fall of 1999 at the Materials Research Society with a high-profile Frontiers in Materials Research Symposium. And after we presented our results, presented this work, about half the researchers in the audience could not believe that number. But there were several people in that audience who said, you know, this has never been tried before, using computations to explore materials. And you guys might be onto something. Well, a year later, some experimentalists at some other universities actually did experiments to find the elastic modulus of nacre in the 20 nanometers size region. And they confirmed what we had predicted a year before. And that gave a big boost to using computations to explore materials. And then subsequently, other groups in the country also started using this technique. But this still did not answer the question why the fracture toughness is so high. And so we scoured the literature, and there were many speculations and theories that some features like mineral bridges, and mineral bridges are important for the growth of seashells. So mineral bridges or surface roughness might be the reason why we have this high fracture toughness. And so what we did was we built computational models of all these different features and tested them computationally and found that none of these contribute to the toughness of nacre. And so basically, we were at a dead end here. We did not know what to do next. But so what we decided to do was to do our own experiments. We said, all right, let's do our own experiments, break the nacre, and look at the fracture surfaces. And then maybe that might give us a clue why the fracture toughness is so high. So what we did was, so our students meticulously carved out the nacre samples from the seashells. And then we pulled those apart and then looked at those samples, put those samples in a scanning electron microscope and looked at the microstructure. When the very first image came on the screen, we just jumped out of our seats. What we saw that these bricks were not just sitting one over another like a brick wall, but each brick was locked into the other brick. So the circles here show you how the bricks are locked into each other. So the bricks are not just sitting one over another, but locked into one another. Now, we wanted to be certain that this is the reason for fracture toughness. So we spent a year building models of these interlocks. And then we did our simulations. And what we found is that because of the progressive failure of these millions of interlocks in this nacre, we get this very high fracture toughness. So basically, what our work showed is that the platelet interlocks are key to the fracture toughness and strength of nacre. And that was an incredible result because people had been trying to understand or to find what's the reason for high fracture toughness, and we were able to find that, and we felt good about it. But while we were doing our experiments, we found something else, which was very interesting. The mortar between the bricks, which was very stiff, it had an elastic modulus 20 gigapascal, so it is a very, very stiff material. And typically when a material is stiff, it is brittle. But here what we found is that not only is that organic mortar very stiff, but it can also deform a lot. It can stretch a lot. And that was fairly a unique property here. And so we decided to investigate this further. Why is it that this is happening? And so what we did was we did molecular dynamics simulations where we pulled first the protein by itself. So the green ribbon which you are seeing being pulled is is the protein, and we're pulling it by itself. And you can see that this green ribbon is stretching a lot, and that's expected. Proteins, they can stretch a lot. And on the left-hand side, the curve, the green curve, shows you the load that's needed to pull that protein. Then what we did was we put the mineral, the calcite mineral, next to the protein, because that's what happens in nacre, that you have this brick which is made of aragonite, and next to that is the protein. So we put the aragonite brick next to the protein, and we started pulling. And as we started pulling the protein, you will notice sort of a jerky motion. You see this red ribbon, which is protein. You start to see this jerky motion, and that reflects as those peaks in the blue graph. So in the blue graph, you see these peaks. Those are because of jerky motion. After that, the water molecules enter in between the mineral and the protein, and then you don't no longer see those big peaks. But what you see is that a much larger load is needed to pull the protein in the presence of the mineral. And that makes the protein very stiff, or the organic phase very stiff. At the same time, what you will notice in these plots is that in both cases, the protein stretches by almost the same amount. And so this sort of answers that question, that why is it that the organic phase is very stiff, at the same time it can deform or stretch a lot. And so basically the mineral proximity, the proximity of that mineral actually influences the protein. And that was a big result that came out of our work. And so basically what we did was we wrote the recipe for the design of biomimetic structural nanocomposite. And with this recipe, now we can design new materials which are light and which are super strong. And so one of the major defense contractors in this country has shown a lot of interest in the use of some of these findings in the design of the hulls for the next generation submarines. We also see the application of these materials for aircraft and spacecraft and cars and bridges and other structural applications. And we thought this was a pretty cool ending to that story. The next nanocomposite material I'm going to talk about is the bone. Now, the bone is a composite material or nanocomposite material made up of collagen, which is a protein, and mineral, which is the hydroxyapatite, say, a calcium mineral. And the beauty about bone is that it has this structural hierarchy. Structural hierarchy means that it has organization at many length scales. So if you see here in this figure, the left-hand side bottom, that's the molecular scale where you have the molecules of collagen, which is the protein, and then you have the mineral hydroxyapatite, which have the size range of nanometers. And so that's a molecular scale. So what happens is that we have an organization that starts from the molecular scale and goes all the way to the whole bone. And that's absolutely beautiful. And what we wanted to do was to find what's the reason for the mechanical properties of bone. What's the origin of these mechanical properties of bone? And in order to do that, we started to first look at the molecular scale. So we wanted to first start with connecting what happens at the molecular scale in bone and how that reflects in the mechanical properties of the bone. And we think that's important if you want to understand bone health in a better way, and also therapies, designing therapies uh, for bone diseases. And so what we did was we pulled the collagen to see in the mineral of the to see if the proximity of the bone mineral has any influence on the collagen properties. So basically what we did was we took the collagen and put it next to the mineral and in all these different orientations which you see in the bone. And then we also compared that with the collagen without the proximity of the mineral. And we did that, and these are the results. And basically what we found that when collagen is in the proximity of the mineral, a much higher load or a force is needed to pull that collagen compared to the situation without the collagen, just like in nacre. Okay, so we have, again, we see similarities between nacre and the bone here. In fact, so this is telling us that the mineral is influencing the collagen in its proximity. Now, up to this point, the collagen and the mineral were always treated separately. This tells us that there's a very intimate relationship between the mineral and collagen, and that needs to be considered. But we wanted to see, so what's the connection between what is happening at the molecular scale and the bone itself? And to do that, we did what's called as multi-scale modeling. Basically, in multi-scale modeling, what we do is to bridge what happens at, in this case, at the molecular scale to what is happening at the next scale, at the fibril scale, and then to the bone. So we build these bridges between these lengths. And we did that, and we found something very interesting. We found that the mechanics of bone is dramatically influenced by molecular interactions between mineral and collagen. To give you some numbers, the effect of molecular interactions on the elastic modulus of the bone, 20% of the elastic modulus of the bone is because of the molecular interactions between the collagen and the mineral. And you would say 20% doesn't seem like a big number. Well, that's the difference between the elastic modulus of a bone of a 25-year-old healthy male and an 80, 85-year-old osteoporotic female. So that 20% is huge, and which means that the contribution of the molecular interactions between the mineral and the collagen on the bone properties is very significant. And so this changes the way we understand bone health and potentially cure bone diseases. While we were studying the bone, we were intrigued by this collagen, which is one of the two components of the bone. And collagen also happens to be the most abundant protein in the human body. Now, at that time, when we started investigating, all the literature, all the work that was being done was on the short 8.5 nanometer long collagen molecule, representative collagen molecule segment. In fact, the actual collagen molecule in the bone is 300 nanometers long. And so what we decided to do was to build a full-length collagen molecule and did molecular dynamics simulation. It's extremely expensive. We need high-performance computers, supercomputers, to be able to do that. But we were able to do that. So let's first see what is collagen. Collagen is made up of 3 strands of proteins. Each strand is a helix. So it has this turn, so it's a helix. And the helix, the helicity in this strand is it's a left-handed helix. Now these 3 helices get together to form this famous triple helix in collagen. Now this triple helix was discovered by Professor Ramchandran at University of Madras. And at one time, it was even called as the Madras helix. This is the structure of collagen, the most abundant protein in the human body. And so what we did was to run simulations for the full length 300 nanometer long collagen. And so we did that. And what we discovered that there was a third level of helicity in the collagen. So basically, this triple helix twists at a pitch of 15 nanometers. So not only do we have this triple helix, but the triple helix itself twists in a right-hand helical manner. And so that was a major discovery coming out of our group on the collagen. But then, like every engineer, so what's the point? I mean, what does that mean? And so we did a lot of simulations, and what we found was that a level 3 helicity contributes to almost half of the total deformation or stretch of the collagen and bone. So that is significant. And up to this point, it was not even known that there was a level 3 helicity there. But the level 3 helicity during the normal day-to-day activities of a human being contributes to half of the stretching or deformation. So the takeaway or the summary of this work on bone is that mechanics of bone is significantly influenced by molecular interactions between collagen and the mineral. Interchain, non-bonded interactions, hydrogen bonds, and structural hierarchy controls collagen mechanics. And level 3 helicity of collagen has important structural implications in bone mechanics. The next nanoscale material I'm going to talk about is swelling clays. Swelling clays are found all over the world, including in the United States. Here, the red regions, the blue regions, and the green regions are where you find a lot of swelling clays. And the red regions are where the swelling clays have the highest swelling potential. These are the worst swelling clays. And you can see right away, we have a lot of these really terrible swelling clays in North Dakota and also on our very campus. And so when swelling clays swell, because when water enters these clays and water hydrates these clays, the swelling clays swell. And because of that, the ground heaves. When the ground heaves, it causes damage like cracks in buildings, cracks in roads, sometimes failures of structures, landslides. So a lot of these things happen because of the swelling of these swelling clays. In fact, the damage caused by swelling clays is of the order of $20 billion every year in the United States alone. So that's huge. And so the problem is that the current soil mechanics theories are incapable of predicting clay behavior. I know it's a bold statement, but that's the truth. And this results in poor design of infrastructure. And this is a challenge for the $10 trillion per year global construction industry. And so because of the magnitude of the problem, we decided this is a challenging problem, we need to work on this. And so what we did was to do a very fundamental study of clays, trying to understand the relationships between the clay fluid molecular interactions and the engineering properties of clays. And in order to do that, we introduced molecular modeling or molecular dynamics in the field of geotechnical engineering. And from that work, what we found is that the molecular interactions between clays and fluids very significantly influence, not only significantly influence, but control the engineering properties of clays. So I'm going to give you one example of how molecular interactions control engineering properties of clays, and that's the flow of fluids through clays. So I'll give you an example of that. And so what you see here is garbage in a landfill. Now, the landfills are lined with what's called as a geosynthetic clay liner. Basically, it's a liner made up of swelling clays. And the point of having that liner is to prevent the fluids in the garbage or in the waste material from flowing through and contaminating our groundwater. So we wanted to see how the fluids, which are typically in a waste, in a landfill, how are those interacting with the clays in the clay liner? And in order to do that, first we had to design a new device because there was no device available to do some very precise calculations of fluid flow for swelling clays or expansive clays. And so on the left-hand top, you see this new device which we designed and fabricated. And then below that, you see a plot which shows the results from those experiments. And I'm going to try to explain those results to you. So on the horizontal axis of this plot, you see the term dielectric constant of fluid. Now, what we found from our molecular dynamics experiments, molecular dynamics simulations, is that as the dielectric constant of the fluid increases, as you go higher towards the right, as the number increases, the strength of the molecular interactions between the fluid and clay increases. So for example, here the water and formamide fluids have very strong interactions, molecular interactions with the clay. On the other hand, if you go on the left side of this axis, when the numbers become small or zero, the molecular interactions between the fluid and the clay become very small or negligible. So for example, when you see TCE and toluene, the dielectric constant is very small, and the molecular interactions between the fluid and the clay is negligible or very small. Okay. Now, on the vertical axis is the term coefficient of permeability. Coefficient of permeability just reflects how fast the fluid is flowing through the clay. There's nothing more complicated than that, how fast the fluid is going to flow through the clay. And if you see these numbers, each of these big grids is a tenfold increase as you go up. So now let's look at the numbers we got. So for water, we found that the coefficient of permeability of the clay was 1 into 10 to the power minus 9 centimeters per second. Now this number is extremely small. Okay, it's a very small number that tells us that the clay is very effective in preventing the water from flowing through. Okay. Now, and that's great, that's wonderful. On the other hand, when you look at toxic solvents like TCE and toluene, actually EPA has called both TCE and toluene as the top 10 most toxic substances found in our landfills. When you look at TCE and toluene, that number jumps up from 10 to the power minus 9 to 10 raised to the power minus 4. It's a huge jump. In fact, if you look on the right, toxic solvents flow 500,000 times faster than water through this clay liner. That's huge. And so what does that number, 10 to the power minus 4 centimeters per second, mean? Well, if you go to a beach, take water and pour it on the sand, what happens? The water simply percolates very quickly through to the sand, and it disappears. Exactly the same thing happens when TCE or toluene is poured on the top of the swelling clay. Basically, the swelling clays in this clay liner are ineffective in preventing these very toxic solvents from flowing through. They're great at stopping water, but they're ineffective in stopping these solvents which have very low polarity, and these solvents don't interact much with clay. So again, the question, why does this happen? And so what you see here are 4 images from a scanning electron microscope, and all these 4 images are for exactly the same clay. They're all identical clay samples. Now, if you see the white line and it says 10 micrometer. So a micrometer is a millionth of a meter. So these particles and other things are very, very small, okay? And that's typical of clay, as expected. So if we look at the left-hand top image, what you are seeing there are relatively large particle sizes, and then you're seeing spaces between the particles or voids between the particles. Okay, those are clearly visible at this scale. Now, let me ask you to take a look at the image on the right-hand side bottom of your screen. And this is where the water has seeped through the clay. And what you see here is that the particles have broken down into smaller and smaller particles, and we really don't see any visible spaces or voids at this scale. Okay. Now, when you go up and look at the same clay through which TCE, which is a non-polar fluid, when that passes through, if you compare that image with the image to the left of it, you will see that those 2 images look very similar. And why do those images look very similar? Because TCE does not interact with the clay, and because of which there is no change in microstructure. And what you see more importantly here in the image on the right-hand side top is you are seeing large void spaces and large particle sizes. So that's why TCE flows 500,000 times faster through the clay than water. And that's the reason for that. So now we also did discrete element modeling simulations, and you can see those void spaces being filled up by broken particles. So the particles which are broken down, they fill up the void space and basically seal off the clay and prevent water and other high polar fluids from flowing through, creating a perfect barrier. So because of molecular interactions, particles break down and fill up these void spaces and seal off the clay. But non-polar fluids which do not have very strong molecular interactions with clay do not break down the particles and do not create this perfect seal. Okay. Now we see the same type of thing happen with compressibility of the clay, the compression of the clay. And we find that clays, depending upon the nature of the fluid in the pores, the clays compress more or they would compress less. And again, that has to do with the molecular interactions between the clays and the fluids. So basically what we have found again here is clay with polar fluids, that is the fluids which interact the most with clays, are less compressible. The clays are less compressible. But if you replace the fluids with low polar fluids which interact less with the clays, you have more compression. So that's another engineering property. An important engineering property in addition to the flow properties is the compressibility. And we see the exactly same thing happening or the strength properties. Okay, so basically what's going on is molecular interactions are changing the microstructure of the clay, and those in turn change the engineering properties of the clay, and also the molecular interactions directly alter the engineering properties of the clay. Now, the engineers, the people who are designing with these clays are used to this clay as made up of three components: air, fluid, and solids. That is conventional. That is conventional soil mechanics. But what we are saying, based on all this evidence, is that clay-fluid molecular interactions are key for accurate prediction of clay behavior. So if you do not consider clay-fluid molecular interactions in clays, you are not going to be able to accurately design with clays. And so molecular interactions control properties of clays, soil mechanics revisited. Now, the last nanocomposite material that I'm going to talk about are polymer clay nanocomposites. When you add small amounts of nanosized clay, we are talking about 1 to 10% or less, small amounts of nanosized clay. And the key is nanosized. That's important. If you add small amounts of nanosized clay in a polymer, polymer could be nylon or polyethylene, the common polymers, there's a 500 to 600% increase in the elastic modulus or the stiffness of the material. So why is it that when you add very small amounts of nanosized clay, we see this huge jump in mechanical properties of the composite? Well, polymer clay nanocomposites were developed by Toyota Motor Company in 1990. And really nobody knew why this happens. And so we decided to take on that challenge. And what we did was, again, did huge amount of simulations. So we got down to the molecular scale, studied the interactions going on between the polymer and the clay and the modifier and all of that. And we also built multi-scale models, basically models that scale different length scales. And from that, we came up with this altered phase theory. Now, it looks like sunny side ups, but this is our altered phase theory. It's one of those situations when you sit down at a desk, you're sitting with a student, and you're discussing something, and you sketch out something, and then that ends up in the paper, and then it becomes something everybody agrees about. Anyway, so the yellow yolks which you see here are actually, those represent the clays. And so what happens is, because of the molecular, and then the light blue is the polymer. And because of the molecular interactions between the between the clay and the surrounding polymer, a large volume of the polymer is altered. The mechanical properties of the polymer in the altered zone is many times more than the mechanical properties of the unaltered zone. So what is going on is, these clays, when they are well disposed, these clays, and again nano-size, actually alter the polymer. And so we call this as an altered phase theory, because these clays are actually altering. And that's the reason why, when you add small amounts of clay and the clays are well dispersed in the polymer, we see this huge jump in the mechanical properties of the polymer clay nanocomposite. So subsequently, what we did was, we took this theory, we built simulation models so that we could design polymer clay nanocomposite materials with tailored properties, with predictable properties for structural applications and so on. But we also decided to use polymer clay nanocomposites in tissue engineering. Now, let me briefly tell you what is tissue engineering. So in tissue engineering, what we do is we take cells from a patient, and then we grow those cells, and the cells proliferate. Basically, they make more cells. And once these cells, we have sufficient number of cells, then we take those cells and then we seed them onto what's called as scaffolds. Scaffolds are simply very porous structures. So it's a material with and a structure with a lot of porosity. Almost 90% is air. So it's very porous. And then what we do, we can give it heat and proper temperature and food, and the cells grow in the scaffold and they make tissue. Now, as the tissue is made, the scaffold starts to degrade. And we want the scaffold to degrade because we want the tissue to basically take the shape and form of the scaffold. And so somewhere in between, we take the scaffold and put it back into the patient. And so now the patient has a tissue made out of his or her own cells. And that is tissue engineering. And so we were working on bone tissue engineering. We wanted to regenerate human bone. And so we were working with polymeric scaffolds. These scaffolds were very soft. These materials were very soft, and we wanted to increase the strength and stiffness of these materials. So obviously, we were working with polymer clay nanocomposites. We said, hey, how about we take the nanoclays and put them in these polymers, and you will have much stiffer and stronger scaffolds. But there was a problem. The problem was the chemicals that were used to make nanoclays were too toxic. So that won't work here. Now, several years earlier, it was just one of those intellectual curiosities about whether clays, the amino acids in the proximity of the clays can make polypeptides or proteins. Okay, there was some speculation, some theories that clays are the reason why the amino acids get together and make proteins. And we said, can we simulate that? And what we found is, yes, it is possible for the proximity of the amino acids is such that you could create the polypeptide bonds and probably make proteins. But something else we found is that amino acids can make nanoclays from clays. Now, amino acids are biocompatible. Okay, so now we have clays themselves are biocompatible. So now we have nanoclays that are biocompatible. So we immediately remembered these simulations which we had done. Actually, in 2007, we published this work, and then we had just forgotten about it. We didn't even think about it. And this is one of those papers that had got a lot of citations, but we just didn't think much about it until we came across this problem. And so what we did was we took these nanoclays using unnatural amino acids. Instead of the natural amino acids, we decided to use unnatural amino acids because they are longer, and our simulations told us that it's better to use those longer amino acids. And so we took those nanoclays and put them in our polymers and made scaffolds out of those. And as expected, our simulations also told that the mechanical properties of our scaffold jumped up by 5-fold. And that was perfect, and that's exactly what we wanted. But then we saw something even more interesting happen. The clays in the polymer clay nanocomposite triggered the mesenchymal stem cells, which are the bone marrow stem cells, to differentiate into the bone cells. That was completely unexpected at that time. And so we got two advantages: increase in mechanical properties, and also that these mesenchymal stem cells or the adult bone stem cells were able to make bone cells. And why is that important? Because those bone cells will then make tissue, the bone, will regenerate the bone. Okay, so that's great. But one of the things we were interested in is that these scaffolds have cells seeded in them, and the cells are making tissue. We wanted to be able to predict how quickly the tissue is formed, how quickly the scaffold is degrading, so that if, let's say, we have an orthopedic surgeon or a patient or a materials person who is designing those scaffolds, to provide them with predictive tools so that they can tell at any given point in time what is going on with the scaffold that's implanted in the body. Whether the tissue has grown, what are the mechanical properties of the scaffolds. So we wanted to provide those tools, and we were able to do that through what we call as multi-scale modeling, where we bridge what happens at the molecular scale all the way to the implant scale. So we are able to do that. Now I'm going to show you some pretty pictures. I think they're pretty. The top picture, the picture on the top, is a picture of the nanoclay scaffolds. Those are the scaffolds. The picture in the middle shows the human mesenchymal stem cells growing on these scaffolds. They're attached to the scaffolds and they're growing. And the bottom right picture is the picture of the regenerated bone when you look at it through an atomic force microscope. And this regenerated bone has the same hierarchy as that of a natural bone. It has the same beautiful hierarchy. It has all of those features. Not only that, the mechanical properties of this bone is the same as that of a natural bone. So basically, we have regenerated natural human bone from human mesenchymal stem cells on our nanoclay scaffold systems, and we also have all of these predictive capabilities in order to do that. And now, of course, this work is going further. The regenerated bone work is going further. We are now going into a translational mode where we are taking these scaffolds, putting them into animal models, and working with some companies on that. Now we have created this beautiful natural bone in the lab, regenerating natural bone. Now this regenerating bone is also a niche for prostate cancer and breast cancer to go to or migrate to. That's metastasis. So both prostate and breast cancer can eventually migrate to bone, and when that happens, it's terminal. At this point, there is no cure when that happens. So we said, all right, these two cancers actually like the regenerating bone environment. How about we put this prostate and breast cancer cells on this, seed them on this regenerating bone while bone is being regenerated? So we did that. And when we did that, we found that we were able to create the tumors and the cancer environment just like it happens in the real life, in the bone. So these were the first test beds for cancer bone metastasis, which came out of our group. And of course, the advantage of this is that we can use it for understanding cancer biology. We can use it for designing new drugs and so on. And we made use of this. So we made use of the tumors from our test beds. And what you see here are the cells within the tumor. And what we are interested was to look inside the cells and study the cytoskeleton or model the cytoskeleton of the cells. So what is cytoskeleton? Just like the spokes in an umbrella. So the cytoskeleton or the skeleton in the cells is just like the spokes in the umbrella, and it's as important. So the health of the cytoskeleton basically defines the health of the cell, and also the cytoskeleton is important for cell migration and other biological features. So in this picture, the green and red colors show two different kinds of cytoskeletal elements. And the blue is the nucleus of the cells. And this is a picture from one of the tumors which we extracted from our test beds. And this is an absolutely beautiful picture. And I think Kalpana was saying once that maybe we should make T-shirts or something out of it. Well, nobody's going to wear it. I mean, this is prostate cancer. So. Anyway, so what we did was to build the first high-resolution mechanistic breast cancer model of cancer cells. So basically, on the left-hand side, you see a cancer cell attached to the scaffold. That's a breast cancer cell. And on the right-hand side is the model, finite element model of our cell. And this cell has all of the features, mechanical features of interest for cancer cells. So it has the cell membrane, it has the cytoskeletal elements like the actin filaments, the intermediate filaments, the microtubules. Then also it has the cytoplasm and then the nucleus. So basically this cell can capture all of the internal structure necessary for the mechanical response of the cell. And what you see down there below, the figure at the bottom, is the simulations where we have taken an indenter, or we are pressing the cell down. Okay, so we are pressing that cell down. And as we press the cell down, we can go inside the cell and see what's happening to the cell, what's happening to all of these different cytoskeletal elements. And so we can get a very good picture of about how the cell responds when it is subject to external stress conditions or loading. And we have basically said or proposed that mechanics as a biomarker for cell progression. So we have proposed for both breast cancer as well as for prostate cancer, we propose mechanics as a biomarker for cancer progression because the individual cancer cells have very different mechanical response compared to the cells in tumors. And so we can use mechanics as a biomarker to see how the cancer is progressing. Okay, so now we go even deeper. We go inside the cell and look at one of the cytoskeletal elements, and that is actin. And why are we interested in that is that we find, left-hand side top is the picture of actin which is spread throughout the cell. So if you take a cell, the actin basically is spread throughout the cell. But when the cells get together to form a tumor, that's the figure on the left bottom, is a picture of cells getting together to form a tumor. The actin migrates to the cell boundaries. So it's no longer distributed throughout the cell, but they move towards the cell boundary. If we are able to capture this actin dynamics, that is the changes to actin from cells to tumors, we'll be able to do a good job with predicting cancer progression. And so on the right, this is just hot off the press. You are seeing the actin molecules being stretched and pulled and twisted, and we're trying to understand the mechanical behavior so that we can feed all of that into what we are seeing on the left-hand side. Now, as engineers, we like to have very powerful prediction tools. I mean, we live by good predictions. If we cannot do good predictions, we'll have buildings that fall and aircraft that crash. So we love to be able to build tools for prediction. And here, our plan is to build computational tools for developing in silico oncology platform for cancer metastasis and treatment. That is the big goal of the things we're trying to do. Well, the takeaway from all of these the research on these 4 nanomaterials is hierarchy, the hallmark of structural biology, as well as structural nuances, influence mechanics of biological systems. And molecular interactions alter phases and control mechanics in nanomaterials, irrespective of the origin, whether it is biological origin, geologic origin, or synthetic origin. And so that's a paradigm shift in mechanics. Well, somebody famous said it was the best of times. But I think it is the best of times, because today we have powerful computers, computational tools, and science to design materials with unprecedented properties to make this world a better place. This was the tale of 4 nanomaterials. And these are the characters of the story. Some of the best and brightest students from around the world who work extremely hard, but they also know how to have fun. And the students who have graduated from our group have gone on to make their own mark in the field. And we are extraordinarily happy with what they have done. So I would like to thank the audience who have joined remotely. Thank you very much for coming. And in this COVID times, taking the time to join this meeting via Zoom. Thank you. And if you have any questions, I'll be very happy to answer those. Thank you very much.
I invite the audience to use the clapping hands and the reactions icon. If you go down to the bottom of your screen and click on reactions, one of the options there is clapping hands. Thank you very much, Dr. Katti.
Thank you.
We'll move now to questions, and I've been receiving a number of questions in the chat sent to me privately. So you in the audience are not able to see these. I'll read these aloud as we can get to them. The first one comes in 2 parts, and it's about nacre. And the 2-part question is this: can the structure of nacre be replicated at the macro scale and result in, for example, materials with high resistance to fracture? Also, are there other natural or man-made materials that exhibit a similar structure?
Okay, so the answer to first question, and that is the quest right now. The quest is to make materials that mimic the nacre structure. Now, what a lot of people have done is to build this brick and mortar architecture at the macro scale, but that does not work. If you look at the key features that are responsible for this high fracture toughness in nacre, is one, of course, is the brick and mortar architecture, but the second is that each brick is interlocked into the other. And the third is the nanoscale feature, that is the soft phase or the organic phase. It has a length scale of tens of nanometers. So all those features need to be present in order to make materials which have fracture toughness or the fracture toughness values that are so amplified. We are talking about thousands of times. If you're looking at fracture toughness improvement by maybe 5 or 10 times, you might be able to do that simply at the macro scale. But if you want to really amplify and use the full power of what we see in Nacre, then you are going to have to do all of those things, that is, have interlocks and have all the nanoscale features in the material that you are designing. And yes, there are several national labs, Department of Defense, and other organizations working on making materials that are mimicking Nacre. Some of them have started doing things in the public domain, and of course, there are some classified in the Department of Defense. But yes, I think now that we have the recipe for how these materials can be made, that problem is not trivial. To take that recipe and to actually make things is not trivial. But today there are, very powerful, robotics and other techniques to be able to design, to make materials with that recipe. The second question, as far as are there other natural materials which behave? And yes, there are many natural materials, especially in the mollusk families, the arthropods and others. Definitely they have. So there are many natural materials. Some of them are of biological origin, which show this kind of amplification in properties.
Good, thank you. We'll jump to another two-part question. When you describe the forces in the interstitial layers in all your material scenarios, are these forces resistive forces, resisting deformation parallel to the layers? And could they be described through the frictional and shear resistive forces at the molecular junctions between the layers? And the second part, could the stem cells in engineered bone form the bone with any scaffold?
Okay, all right. So let me first answer the first question. So at the molecular scale, when we are looking at, so when we start looking at, so what is friction at the molecular scale? So what we are seeing is that it is the non-bonded interactions. So between the atoms of one phase interacting with the atoms of other phase, that's one. And also bridging in between are the water molecules. So when these two materials are sliding past one another, the non-bonded interactions between the atoms of one phase, the mineral, and the other phase, the protein, and the bridging water molecules in between, all of them together provide resistance to the flow of the protein as we pull the protein. So it's not so-called conventional friction, but it is at the atomic or molecular scale, it is the non-bonded interaction. So you don't have these the frictional features which you see in conventional friction, but rather you have these non-bonded. So you have the electrostatic and van der Waals interactions between these atoms that are responsible for providing these resistance to sliding or movement. So that is the other question I'm trying to remember. What was the other question? The second part?
The second part asks, could the stem cells in engineered bone form the bone with any scaffold at all?
Okay. So if you put these, so there are many different materials to make, to engineer tissue. So in the field of tissue engineering, there are many different materials. The materials vary from ceramic materials to polymeric materials, even metal foams, or even using the bone itself as a scaffold. So basically you remove the organic phase of the bone and then use that as scaffolds. So yes, there are many different materials that could be used to make bone. In our case, we selected the polymeric materials and also seeded those with nanoclays, because then we have very good control on the mechanical properties of the polymer. And also another important thing is, unlike ceramic materials, the polymeric materials can degrade and dissolve when the tissue is completely formed. That's one advantage. And second, which like I mentioned, it was unexpected, was that these nanoclays are able to trigger the stem cells to make different bone cell lines that make tissue, which is fairly unique when it comes to the material system which we are using. So if you use scaffolds which are not the system, the nanoclay system we are using, you will need growth factors or differentiating media in order to get the stem cells to differentiate and make the bone material, the bone.
Very good, thank you. There's a question about collagen. Does the third level helical structure of collagen occur only in bone, or does it occur in other tissues with abundant collagen? Let's see, this has moved on me. Let me find the rest of it. Does it occur in other tissues with abundant collagen like skin or blood vessel walls?
You know, that's a good question. And I think we'll write the next NIH grant for that. We know for sure that it happens in bone, but we do not know because as a scientist, I would not speculate. I do not know the answer. We do not know for other material systems, other, blood vessel walls or other tissues, whether you have the third level of helicity or not. I do not know, but that's a very good question. I'm going to make a note of it to write the next grant to NIH.
Very good, thank you. There are two more questions, and I'll end with Dean Kessler's, but before we get to that, there's a question that about the four materials you have described. 3 are inert, but the bone is human and a live organ. Why did you select bone? And does it have any special features similar to inert materials?
Well, I wouldn't call nacre as an inert material. Nacre is a natural material which grows. So in that sense, so basically we are looked at 2 biological materials. Yes, the bone is, is a regenerating material, but so is nacre. So these are both biological materials. Now, why did we select bone? Okay, there's a story for that. And the story is that, so we were working at the time on nacre, and we saw that the nanoscale, at the nanoscale, the proteins were strongly influenced by the proximity of the mineral. Now, at that time, Kalpana was working on new materials for joint replacement. So while she was doing that, so we were discussing, again, I think that's fairly typical over dinner table, you sit and discuss science, but we were discussing about the human bone, and human bone is also has the similar nanoscale features as nacre. And since we saw this amazing properties of nacre because of the proximity between the mineral and the protein, we thought maybe that's what's going on, that could be going on in the bone also. And so we decided to then do what we were doing for nacre, to do something very similar for bone, and we found that bone was also showing us the same kind of a behavior as nacre. So basic, and then the clays, we have always been working on clays for a very long time, and then polymer clay nanocomposite was another synthetic material. But the key point here is that in all these cases, whether it was a natural, the biological systems, or geologic systems, or synthetic material systems, in all these cases, if you have nanoscale features in your material, then the mineral proximity or the molecular interactions basically are going to have a very significant effect on the properties of the material. So that's a big takeaway from this. Now, we have also done work on oil shales. I didn't have time to talk about oil shales. So we have done work on oil shales also. Again, that we are looking at the proximity of the mineral, that is the shale, and the oil molecule. So basically, this is happening. This is a nanoscale phenomenon, irrespective of what the origin is.
Good, thank you. Before the presentation of the award, we'll end with a question from Dean Kessler. For the NACA research, you mentioned that the platelet interlocks are key to high fracture toughness, with different stress levels contributing to different partial interlock failures. How is the fatigue resistance for NACA as a result of this? Let's see, how is the fatigue resistance for NACA as a result of this cumulative failure mechanism that could occur at lower stress levels?
Mhm. Yeah, no, I think that's a very good question. And again, when we look at the fatigue mechanisms, we have to basically look at look at those mechanisms. So we have not looked at the fatigue mechanisms. What we have looked at is the fracture toughness. Now, again, when we talk about fatigue mechanisms, it's going to be also be related to the not just the structure of NACA, I mean, not just to the structure which we are mimicking, but also the material properties of the constituents. So, for example, are we using, high strength ceramics to replace aragonite or the calcium carbonate, or are we using polymers or something else as the in place of the organic phase? So it will all depend on that also. So we can sort of control some of the fatigue behavior, but we have not studied fatigue behavior in NACA. So when we are mimicking the structure of NACA, the focus has more been on the fracture resistance. And then when you're mimicking that and you want to also have a fatigue resistance, then you'll have to bring that into play by choosing the right materials for that.
Well, thank you very much for so much research throughout your career and all the good applications it's been put to. We need to move on, even though some other questions have come in. And I invite you to email your questions to Dr. Katti if we didn't get to them tonight. At this point, we'd like to make the presentation of the award. And for this, President Brashani is in the Century Theater, fully masked. And I will turn it over to him at this point to present Dr. Katti with the Faculty Lectureship Award.
Well, Dr. Katti, thank you very much for an absolutely outstanding presentation. Aside from those joining us here in the theater today, obviously over 70 of your former students, most of whom are now colleagues from around the nation, and in fact, the world joined us this evening for an outstanding presentation of your work on this topic. It was a pleasure to be here in person, but it's also impressive when we recognize that those over 70 people are scattered literally around the world. Many of them are up in the middle of the night or the wee hours of the morning because of their passion for your field, as well as their support of your work and their ties to you and the inspiration that you've provided them. Truly exciting to be a part of the creation of knowledge and the sharing of knowledge that really only happens in a university setting. And that's what makes this particularly exciting. Not only is this a highly competitive award, but it's also recognizing scholarship in its absolutely finest form, not just the creation of new knowledge, but again, the sharing of that new knowledge, and outstanding opportunity and quite an accomplishment. And my congratulations to you. In the style of the day, we're not going to hand you something here this evening. So it's in a sense as a narrative virtual awarding, but nonetheless, the recognition is absolutely as important and significant as if we were handing you a plaque with your name on it right now. But thank you for an outstanding presentation. Thank you to the people in the audience, and again, literally around the world, who have joined us this evening for this presentation, and frankly, this recognition about standing scholarship. The creation of new knowledge may be one of the most grand endeavors that anyone can pursue as a career, and the presentation this evening I think really exemplifies the nobility of what we do at a research university and the nobility of the people that are doing that at a research university, the creation of new knowledge. I can't imagine a greater accomplishment. Thank you, Dr. Katti. Thank you to our audience this evening, and I'll turn it back to Dr. Maleth to bring us to closure.
Yes, thank you, President Brashani. And this brings us to the end of this year's awards ceremony for the faculty lectureship. And I want to thank Dr. Katti in particular for such wonderful research and for sharing it with us tonight. If you do have further questions, I invite you to email those to him, and I invite you as well to send your own individual congratulations to Dr. Katti by email or by other means if you have those means to connect with him. Thank you for participating tonight, everyone in the audience. We hope to have you back for the next faculty lectureship, which will be the 60th. Farewell till then.